Hello everyone, welcome back to my video channel. In the previous video, we learned or we discussed about monosaccharides. Okay, so if you didn't watch that video, if you are new to this channel, if you want to learn about monosaccharide, you can go back to that video and watch it. Okay, thank you. And now in this video we are going to learn about oligosaccharides, disaccharides and polysaccharides. First we will learn oligosaccharides. The definition that I have wrote here is carbohydrates that releases 2 to 10 monosaccharide units on hydrolysis are called as oligosaccharides. Okay, on hydrolysis if they releases 2 to 10 monosaccharide units, those are all sugars comes called as or comes under the oligosaccharides. And depending upon the number of monosaccharides released, oligosaccharides can be further divided into 4 types, that is disaccharides, trisaccharides, tetrasaccharides and pentasaccharides. So as in the name indicates, di means 2, okay, which releases 2 units on hydrolysis. Okay, tri means 3, which releases 3 molecules on hydrolysis, and then tetra means 4, which releases 4 monosaccharide units on hydrolysis, and then pentasaccharides, penta means 5, which releases 5 units on hydrolysis. Now first we will learn the first one, that is disaccharides. So as I said just now, di means 2, okay, which releases 2 molecules on hydrolysis. But here the definition that I have wrote is quite opposite to the definition of the which I have given here. Okay, means it is just opposite to that. Means when 2 monosaccharides units combines, okay, they through the glycosidic linkage that forms a single unit, that one unit, okay, di means 2 molecules combine to form a single molecule. Okay, you can say in the opposite direction also. So I am saying in this direction. So the definition that I have given here for this is a carbohydrate, right? So on hydrolysis it is going to release the 2 units. You can look at in the diagram here, I mean the equation that I have wrote here, watch it here carefully. So this is the carbohydrates. On hydrolysis it releases the 2 molecules. Okay, we can define in this way also, and again you can define in this way also. Means the glucose is a carbohydrate, right? This is the monosaccharide, and this is another glucose, that is another glucose monosaccharide in it. These two, 1 plus 1, 2, di, so combine to form a carbohydrate. Okay, that forms the disaccharide. So if I had to say in a sentence form, a disaccharide is the sugar formed when 2 monosaccharides are joined by or linked by glycosidic linkage. So I have given in this way, that's why I wrote this one in the opposite way. Okay, both are same. I mean the definition for make you to understand, the way I am trying is correct. Okay, now we will move on to the next one. These disaccharides are simple sugars and are soluble in water. Examples, now we will look at the examples for disaccharides. So the first one, glucose plus glucose on hydrolysis, means water is going to release here. So it forms the maltose. So where we can find out the maltose? In fruits, okay, in sugar beets, okay, there we can find out the maltose. Then glucose plus galactose on hydrolysis gives rise to, water is going to release and it forms the lactose, that is we can find out in the milk, okay, milk sugar you can say. Next example, glucose plus fructose on hydrolysis, which releases the water, which gives rise to sucrose. So you can find it the sucrose in cane sugar, what we call it as a cane sugar cane. Okay, now again this disaccharides divided into 2 types, that is homodisaccharides, the second one heterodisaccharide. First we will look at the first one, homodisaccharides. Homo means similar, okay, hetero means different. Homo, first we will look at the homodisaccharide. Homo means homodisaccharides are nothing but carbohydrates only. On hydrolysis, a single kind, as I mentioned before, single means same kind, similar kind of monosaccharides on hydrolysis, that forms the homosaccharide. For example, If you take the maltose here on hydrolysis, glucose, both are similar, right? So same molecules, monosaccharide units are going to release, same molecules are going to release. So that's why maltose comes under the homodisaccharide. The next one, hetero means different. Disaccharides which gives two different monosaccharide units on hydrolysis, those comes under the heterodisaccharides. Example, lactose on hydrolysis which gives the glucose plus galactose. Here you can look at the monosaccharide here. The glucose is one different monosaccharide and galactose is another different monosaccharide. These two molecules are different. That's why these are heterodisaccharides. Di means two molecules. So I hope you understood this. Now we will move on to the next type. So as I mentioned before, tri means 3. Okay, we are learning about the oligosaccharides. Oligosaccharides again divided into these 3 classes. We completed with the disaccharide. Now we are moving on to the trisaccharide. As I mentioned before, trisaccharide, tri means 3 monosaccharide units are going to release. Example for that is raffinose. And the next one, tetrasaccharides. Tetra means 4 monosaccharide units are going to release, and the example for that is statiose. Now the pentasaccharides, penta means 5 monosaccharide units are going to release. Example is verbascose. For example, if raffinose, I said 3 monosaccharide units are going to release. I am giving one example here. Raffinose, what are the 3 monosaccharide units are? That is glucose, fructose and galactose. Okay, we will know the more points on disaccharides here. The disaccharides are the most common oligosaccharides. Okay, and then sucrose is the most common disaccharide, and it is the transporting sugar in plants. Okay, you have to remember this point, it is very important, and it is taken as a standard for indicating the sweetness, and the sweetness of sucrose is taken as 100, and in the same scale the sweetness of fructose is 180. And one more carbohydrate, that is trehalose. It is the transporting sugar in insects, and it is found in hemocoel of the insects. And remember, these points are examination points, like in a competitive exam they will ask these questions. So you have to remember these questions. Okay, and this is the structures which I have wrote here, nothing but for disaccharides only. That is glucose plus glucose forms the maltose. Okay, this is the glycosidic linkage. Okay, and this is the pyranose ring. Okay, and the next one, glucose plus galactose. Okay, which gives rise to lactose. Okay, this is also a glycosidic, another type of glycosidic linkage. And then the next one, glucose plus fructose, and which forms the sucrose. Okay, and here you can see fructose. It's actually not the pyranose ring, it is a furanose ring. Okay, as it is a ketone, and these are the aldehydes. Now we will move on to the next one, that is polysaccharides. Polysaccharides have another name. These are also called as glycans or non-sugars. So what are these polysaccharides are? Now we will learn in more detail. Poly means many. Okay, here in case of this carbohydrates, the monomeric units will be many. Okay, it may be more than 1000, more than 100. Okay, the monomeric units are more. In case of oligosaccharides, it is only 2 to 10 monosaccharides units. Okay, here the polysaccharides are highly complex carbohydrates and are most abundant in nature. And as I used to mention, carbohydrates are like monosaccharides, they are sweet in taste, but the polysaccharides are tasteless carbohydrates, and they are made up of branched and unbranched linear chain of monosaccharide units. Okay, they are made up of branched and unbranched linear chain of monosaccharide units, like this. If I have to make you to understand, so it is if you think this is the polysaccharide, okay, and this is it is unbranched. Okay, if you think this is the polysaccharide, okay, and these are the monomeric units. I am just drawing a rough diagram just to make you understand. Okay, so these are the if you think these are the monosaccharide units, and these will be more than 100. Okay, more than 100 to 5000. Okay, and this is the unbranched. Okay, it doesn't have any branches, and if you take this one from here, you are going to find the another one like this. Okay, branches like this will be present. Okay, so this will become the branched polysaccharide. If I have to give the definition here, polysaccharides are carbohydrates that releases more than 10 monosaccharides units on hydrolysis, or you can define in another way, the polysaccharides are polymers of monosaccharides units joined by glycosidic bond. Okay. Now coming to the general formula for polysaccharides is C6H10O5 whole bracket N. This is the general formula for polysaccharide. See here, the general formula for disaccharide is this, that is CnH2O N minus 1. This is the general formula for disaccharide. For example, here I have taken the example of maltose, sucrose and lactose. So according to that, here monosaccharides we have only C6, right? In monosaccharides C6H12O6, okay, glucose. So 6 carbon atoms will be present. In di means 2, it will be double. That's why C12, okay, and then H2O, here the N is 12, so that's why 12 minus 1. So next step, C12 H2O, 12 minus 1 is 11. So when you do the multiplication to this, C12 H22O11. Okay, this is the general formula for maltose, sucrose and lactose. Okay, you have to remember this formula for the disaccharides. Coming to the polysaccharides, as we are discussing the polysaccharides now, the general formula for polysaccharides is CxH2O whole bracket Y, or another formula is C6H10O5 whole bracket N. Okay. So where here X indicates the large number of monosaccharides units. Large number of monosaccharides units means, as I said before, it may be more than 100, more than thousands. Okay. Where here for this formula N indicates, that's why the number of monosaccharide units. Okay. So this is about the general formula of the polysaccharide. Now they have more than 100 subunits, okay, because in case of branched ones we can get the another subunits as well. So they are very having the high molecular weight and are insoluble in water. So but disaccharides are soluble in water, but polysaccharides are insoluble in water. And they have right end is called as the reducing end, and if you look at here, as I explained, this one is polysaccharide, okay, and here the right end is called as reducing end, this end is called as reducing end, okay, and the left side, sorry, this is the left side, right, this is non-reducing end, okay, and this is the right side, right, okay, this is the reducing end, okay. So I guess I have wrote here itself, okay, and here they acts, and what is the functions here, they act as a storage molecules, okay, the polysaccharides acts as a storage and structural molecules. Structural means they will give the support to the structures or organs, okay, and they act as a storage molecules as well as the structural molecules. And again these polysaccharides are divided into or classified into two types, okay, that is homopolysaccharides and heteropolysaccharides. I guess I have explained the same thing, that is the homodisaccharides, heterodisaccharides. The same definition goes for here, but these are polysaccharides, okay. These homopolysaccharides yield only one type of monosaccharide units on hydrolysis, that's why these are homo means similar, that's why these are homopolysaccharides. For example, starch, cellulose, glycogen, dextrin are the examples. For example, if you have to say one, if I have to give one example, starch, it will see, this is maybe not visible, I just wrote in the pencil, if it is visible that's quite good enough, okay. So starch, it has the similar monosaccharide units, that is glucose. So like this, there will be more than 100 like that, okay, more than 100, more than 1000 like that. Now coming to the heteropolysaccharides, here the heteropolysaccharides yield more than one type of monosaccharide units on hydrolysis. As I mentioned before, hetero means different, okay. Example for that, heparin, hyaluronic acid and hemicellulose are the examples. For example, heparin is going to release like different monosaccharide unit. The one monosaccharide unit is glucuronic acid sulfate, and then another molecule is glucosamine sulfate, and hyaluronic acid going to release glucuronic acid and N-acetyl glucosamine. So if I have to give some of the examples here, I have made it a clear is important monosaccharide and homopolysaccharide units, and also heteropolysaccharides and monosaccharide units. Okay, so homo means it is going to release only one similar molecule, right? So the polysaccharide, that is homopolysaccharides, are like cellulose, starch, amylase and amylopectin. These have a chain of or similar monosaccharide unit, that is glucose. And the next one, inulin. This is the monosaccharide units for the inulin is fructose. And then for chitin, the monosaccharide unit is N-acetyl glucosamine. And for the pectin, it is galactonic acid. Okay. Now we will move on to the heterosaccharides. For heterosaccharides here, the capsule of Diplococcus, the bacteria, okay, or Streptococcus, the monosaccharide unit is glucose or glucuronic acid. Here, as I mentioned before, hetero means different. They have to release the different kinds of monosaccharide units. That's why these are different. Here, the single one, same monosaccharide units. The long chain of fructose will be present. Here, the different, that is glucose and glucuronic acid. The next one, the peptidoglycan. It is also called as murin. It releases N-acetyl, or it is composed of N-acetyl glucosamine and N-acetyl muramic acid. The third one, hyaluronic acid. It releases glucuronic acid and N-acetyl glucosamine. And the next one, chondroitin, glucuronic acid and N-acetyl galactosamine sulfate. The next one, dermatan, hydrochloric acid and N-acetyl galactosamine sulfate. Keratin, galactose and N-acetyl glucosamine sulfate. You have to remember this. There is a slight vary here. This is galactosamine. Okay, this is glucosamine, and I have just given the important ones that are frequently asked in the competitive exams. So it is better to remember this one. Okay, I have chosen and I have given the examples here. Heparin, glucuronic acid sulfate and N-acetyl glucosamine sulfate. Okay, these are the example for homopolysaccharides and heteropolysaccharides. Now we will learn the few more points on polysaccharides. So we can classify the polysaccharides in another way also. So these polysaccharides again divided into three types, that is food storage polysaccharides, structural polysaccharides and mucopolysaccharides. Here the food storage polysaccharides means they are going to store the food, okay, and the structural polysaccharides means they will support the structure or the like cell membrane, like that, okay. So the example for food storage polysaccharides are starch, inulin, glycogen. Structural polysaccharides examples are chitin, cellulose, pectin, peptidoglycan and agar. And coming to mucopolysaccharide, muco means mucus, like a liquid, like fluid like content is going to release, for example on our joints. So that liquid is very important, if you take the joints, so that will give the flexibility, that mucus, okay, that will help to move the leg, okay, so we don't get any pain. So because of these mucopolysaccharides, so these should be present in our body. We should take the polysaccharides to the diet, so that mucopolysaccharides to the diet, so that it will secrete the mucus like content that is very useful for the joints, okay. Example for this is hyaluronic acid, heparin, keratin sulfate, dermatan sulfate and chondroitin sulfate. And few more important point on the polysaccharides are, here the cellulose is the most abundant biomolecule and it is a polysaccharide, okay, this is the most abundant one, and the chitin is the second most abundant polysaccharide. The first one is cellulose and the next one is chitin. And I guess I have wrote the definition here for the mucopolysaccharides, because these two saccharide definition is here. The mucopolysaccharides are long chain of sugar molecules that are found throughout our body, and they are often in mucus or in fluid around the joints, okay, and they are most commonly called as glycosaminoglycans, that is GAG. Okay, remember this GAG, this question on this GAGs, the question is asked in the competitive exams. Okay, so in higher studies, if you go to the higher studies, this point is very important. There we are going to learn in depth. Okay, remember GAGs, okay, glucosaminoglycans. Now some of the important biological importance of the polysaccharides here. The heparin is going to act as the anticoagulant, which is found in the liver, spleen and lungs and blood. Okay, it is going to act as an anticoagulant. And the next one, hyaluronic acid, is found in the umbilical cord. Okay, so umbilical cord and in the vitreous humor, means it is the space between the lens and retina of the eye. and in the joints and connective tissue also as binding materials, like this, as a binding material in animal cells, and it has a lubricating action also. Um, binding material in animal cells, and it has a lubricating action also, and in tissues it forms an important part of the cementing ground substances. Okay, that's about the polysaccharides. In the next class we will study about the reducing sugars and non reducing sugars, and then we will learn about the learn about structure and also what are the stereoisomers are. Okay, I hope this video is helpful for you all. Thank you everyone.